Hello students, this is Kanika. Welcome to my channel PrepBiology. So by continuing with the Unit 1 of Class 12th Biology, that is reproduction, we have started chapter number 1 of this unit, that is sexual reproduction in flowering plants. This is class 8th of this series, and in this lecture we will understand about the process of Megasporogenesis and the structure of the female gametophyte. Let's start with the lecture. If we talk about the Megasporogenesis, as the name suggests, Megasporogenesis, Megaspore plus genesis. Megaspores means the large spores, and genesis means process. So, the formation of large spores or megaspores, that process is called the megasporogenesis. We know that there are two types of the spores in plants, that is the microspores and megaspores. Microspores are the male part and megaspores are the female part. Okay, so in this lecture we will understand about the different stages of the megasporogenesis which occurs in the female reproductive part. Okay, if we see the megasporogenesis, we know that there is a ovule which forms the female gametophyte. Okay, we have already covered in the previous lecture that the structure of ovule, this is the structure of the ovule, and at the middle of this we have embryo sac. We have covered this part. Okay, now inside the embryo sac, the development occurs. Okay, so in the embryo sac, the megasporogenesis occurs. Okay, and we know that there are two poles present in the structure of the ovule. This pole is called the micropylar end, and the basal end is called the chaliceal end. Okay, so let's start with the different stages. The megasporogenesis is the formation of the megaspores. Okay, now the megaspores, this is the first stage in which we know this is the embryo sac, and inside the embryo sac the development occurs. Okay, now we know that around the embryo sac there are the mass of cells, that is called the nucellus. These are the different mass of cells present around the embryo sac, that is called the nucellus. Okay, inside the nucellus, make sure, this is important, at the micropylar end, the ovule generate or differentiate a special kind of cell, that is called the MMC. MMC, what is the full form of that? That is the megaspore mother cell. At which end megaspore mother cell generate? At the micropylar end. This is very important. Okay, now what are the different features of the MMCs? At the micropylar end, the special kind of cells generate in the ovule, that is the MMC, that is the megaspore mother cell. It has two main properties. First one is that it has the dense cytoplasm and well developed prominent nucleus. Okay, at the middle you all can see here it has prominent nucleus, and around the nucleus it has the dense cytoplasm. These two features are very, very important of my megaspore mother cell. Now, different divisions occurs in the megaspore mother cell. The first meiosis occurs and this cell, megaspore mother cell, will convert into the dyad. That is the two celled. This is one and this is two. Okay. Now meiosis 2 will occur. Meiosis 2 will occur and these two dyad megaspore will develop into the 4 megaspore. This is 1, 2, 3 and 4. 4 megaspore structure that is called the megaspore tetrad. Okay, this is somewhat similar to the microsporogenesis. We already have discussed that process in the previous classes. Okay, this is the megaspore tetrad. Okay, now here the 2 points are very important, that which kind of divisions occurs in the megaspore mother cell or MMC, that is the meiosis 1 and meiosis 2, that is meiotic division occurs. Okay, so there are 4 megaspores are formed, that is called the megaspore tetrad. Now we have the 4 megaspores formed and each have well developed nucleus in it. Now out of 4, this point is very clear, listen carefully, out of 4, 3 cells are degenerate. Okay, out of 4, 3 cells of the megaspores degenerate, 3 cells degenerate and only 1 megaspore is functional. So out of 4, 3 megaspores are degenerate and only 1 megaspore will functional. Okay, so that 1 megaspore then further develop into the female gametophyte or embryo sac. So here one term is very important, that the development of the female gametophyte or the embryo sac from only 1 megaspore, single megaspore, that development is called monosporic development. Mono means single and sporic means spore. Okay, so from the single spore, from the single megaspore, the development of the female gametophyte occurs or embryo sac occurs, that development is called the megaspore development. This is all about megasporogenesis. I hope these all stages are clear to you all. We have covered the megasporogenesis, that is we have one functional megaspore is here. Okay, so these are the megaspore tetrad. Out of 4 cells, 3 cells degenerate and only 1 megaspore is functional. Now, the female gametophyte, that is the embryo sac, the development of the embryo sac occurs from this step. We have 1 megaspore. Okay, now in the 1 megaspore, the mitosis will occur. This point is very clear. In megasporogenesis, we have meiosis occurs, meiotic division occurs, and in development of the female gametophyte, that is the embryo sac, which division occurs? Mitosis will occur. Okay. Now the nucleus will divide into 2 nucleate stage. Okay. Okay. So that stage is called the 2 nucleate stage. And the nucleus are shifted towards the pole. This is one pole and this is the second pole. Now, in 2 nucleate stage, another mitosis division occurs. It will form this 1 nucleus change to 2 and this 1 nucleus change to convert it to 2 nucleus. So total we have 4 nucleate stage. In 4 nucleate stage, another mitosis occurs, division occurs, and we have these two nucleus will change, converted to 4, and these 2 nucleus converted to 4. So 4 plus 4, 8 nucleate stage we have. So in the development of the female gametophyte, that is embryo, the mitosis occurs. Okay, so this point is very important. Now we have 8 nucleate stage. Okay, now these 8 nucleus will will shifted or arranged in the manner that six nucleus or the 6 cells are at the shifted towards the pole, and two cell or one cell or the two nucleus will at the arranged in the center. So we know that we have the chaliceal end, we have the micropylar end. Okay, so at the chaliceal end, the out of 8 cells or 8 nucleus, we have three cells arranged at the chaliceal end, and the 3 cells are shifted towards the micropylar end. And 2 cells or the 2 nucleus will at the center. Okay, so this is the arrangement of 8. This is the arrangement of the cells occurs in the female gametophyte. Now, if we see the structure of the mature embryo sac, okay, so this is the structure of the mature embryo sac. We have discussed the micropylar end and the chaliceal end. There are 2 ends in the structure of the ovule. At the chaliceal end, the three cells are present, and each cell have their cell wall. Okay, so each cell have their cell wall. So at the chaliceal end, the 3 cells which are present, that is called the antipodal cells. Okay, so make sure that the antipodal cells are present at which end? Chaliceal end. Now, if we talk about the micropylar end, the 3 cells present at the micropylar end, and these cells, out of 3 cells, the 2, this is 1 and this is 2 second, the 2 cells are called the synergid cells, and the 1 large cell, that is called the egg cell. Okay, so in the micropylar end, we have 2 synergid cells and 1 egg cell, and they will form the egg apparatus. Okay, so in the egg apparatus, we have 2 kinds of cells. One is egg cell and second one is the synergid cells. The number of synergid cells are 2 and number of egg cell is 1. Okay, this is at the micropylar end. So now at the center of this, we have central cell present. Central cell present. So central cell have 2 polar nuclei. Okay, so central cell have how many nucleus? 2 nucleus. This is 1 and this is 2. So these nucleus are called the polar nuclei, or okay, so these nuclei are called the polar nuclei. Now, if we see at the chaliceal end, we have 3 antipodal cells. And at the micropylar end, we have 2 synergid and 1 egg cell. And 2 synergid cell. Okay, and at the middle we have the central cell, and central cell contains 2 polar nuclei. So this is the arrangement of the cells in the mature embryo sac. Now, one point is very important here. We have here at the micropylar end, the 2 synergid cells are present. Inside the 2 synergid cells, this is 1 and this is 2, we have very important cellular thickening. Cellular thickening is present thickening is present, that is called the filiform apparatus. Okay, filiform apparatus is the special kind of the cellular thickening present in the synergid cells. The function of the filiform apparatus is it will guide the pollen tube. When fertilization occurs, it will guide the pollen tube into the synergid. Okay, so this point is very important. What is the function of the filiform apparatus which is present in the synergid cells? It will guide the pollen tube into the synergid during fertilization. Okay, so this is the mature embryo sac. We have discussed different arrangement of the different cells in the mature embryo sac. Now, the stage, a very important stage, that is the 7-celled 8-nucleate stage. This question is very important in your board exams. This question is repeatedly asked in your previous years. So what is 7-celled 8-nucleate stage? 7-celled. First understand this 7-celled. 7-celled is, we have at the chalice end how many cells? This is 1, 2 and 3. 3 cells. And at the micropylar end how many cells? 1, 2 and 3 cells. And in the middle of the mature embryo sac, how many cells? Only 1 cell is present. Central cell, that is central cell. So 3 plus 1, that is 7-celled. Okay, understood? Now 8-nucleate. Understand it carefully. 8-nucleate. 8-nucleate means the antipodal, each antipodal cells have their nucleus. 1, 2 and 3 nucleus here. 2 synergid cells and 1 egg cell have their nucleus. 3 nucleus here. And inside the central cell we have how many nucleus? 2 nucleus here. 3 plus 3, 6 and plus 2, 8 nucleate. So this stage of the mature embryo sac, when these all cells arranged in this manner, that is 7-celled 8-nucleate manner, that stage is called the 7-celled 8-nucleate stage. This stage is very, very important. Understand it carefully. Okay, if we see this diagram, this is the diagram is from your NCERT biology textbook. We have discussed these parts, that is the Megasporogenesis, we have one megaspore mother cell at the micropylar end. That meiosis will occur, it will divide it into 2, that is the megaspore dyad, and then another meiosis occurs, then megaspore tetrad will form. Out of 4 cells, 1 cell will functional, from one megaspore mother cell, the development of the female gametophyte occurs. That development is called the monosporic development. Now, in the female gametophyte, we have this one megaspore. The mitosis will occur. It will goes to the converted to the 2-celled stage, 4-celled stage, and 8-nucleate stage. Okay. Then these all cells will arranged. We have 2 ends, chalizal and micropylar end. At the chalizal end, 3 antipodal cells are formed, and at the micropylar end, the egg apparatus is formed. In egg apparatus, there are 2 kinds of cells, egg cell and synergid cell. In the synergid cells, special kind of thickening is present, cellular thickening is present, that is called the filiform apparatus. Its function is to guide the pollen tube into the synergids during the fertilization. Now at the middle we have the central cell. central cell contains the two polar nuclei. That stage is called the seven-celled, eight-nucleate stage. So this is all about the Megasporogenesis process and development of the mature embryo sac, that is the female gametophyte. This is all about today's lecture. I hope this lecture is beneficial to you all. If you like this lecture, like, share and subscribe my channel, that is PrepBiology. Keep studying, study hard, study smart. Thank you so much.